Hi, I'm Michel Thibault de Chaten, and thank you for watching Parcellating the White Matter with Functional Neuroimaging. You probably came across our recent publication where we directly map terms derived from a task-related functional MRI onto the white matter. This is important because for the past 2 decades, a lot of interest have been attributed to the way the brain is connected. For instance, I published myself an atlas of human brain connections, but you could also be interested in all the works that Oliver Spohn has been doing in his books and looking at how the brain is connected using the graph theory. In Cerebral Cortex, a book that has been published with Deepak Pandya, Mike Peptrides, and other authors, you'll be able to explore how connections and the brain cytoarchitecture has been evolving. In Fiber Pathway of the Brain by Schmaman and Pandya, you'll be able to see how the monkey brain is connected using axonal tracing. In I Am My Connectome, you'll have the theory behind why the way the brain is connected can define who we are. With a book from Alex Fornito et al., you'll have an explanation of the means and the method how to study brain connection using graph theory, and with the books that will come out very soon, later this year, The Entangled Brain, you'll be able to understand better the importance of connections for the functioning of the brain. But besides this book, further reading can also enlighten you into why connections are important for the function of the brain. And if you read the work of David Van Essen, he says that area can be arranged in well-defined hierarchy on the basis of the pattern of interconnections. Or if you are more interested about the work of Marcel Mesulem, he says that nothing defines the function of a neuron more than its connections with other neurons. Finally, if you're more a Carl Zille's person, you would be able to read in his writings that afferent, efferent, and intrinsic connection, as well as cellular type and their properties, are the structural basis of a brain region's function. So connections are very at the core of the functioning of the brain. So why don't we study the function of white matter connections? In order to do that, we decided to use an approach putting in our advantage the base data that we could put our hand on. And those are all open free data that you can use yourself. On the one hand, we wanted to define the functional organization of the cortex by using meta-analytic map derived from the website NeuroSynth. NeuroSynth is an automatic website that will collect coordinates of functional activation and create meta-analytic maps for you to download and analyze. In terms of how the human brain is connected, we chose to use the Human Connectome project using data at 7 Tesla because there was the highest resolution available when we did this project. The Human Connectome project provides you with data, but the tractography itself of the data is not computed. And for that, we use the software StarTrack that we've been using for almost 2 decades now, just because we tune this software to what we can see in post-mortem dissections. And that's the main reason why we're using it, is actually finely tuned to what you can do using post-mortem dissections of the white matter. Finally, in order to simplify a little bit how things are connected together, we used the current best parcellation that we have of the brain, which is a multimodal parcellation by Mike Lasser, published in Nature in 2016. Then once the processing of the data was done with StarTrack, the idea was to collect as much information as possible in the way the brain is connected. And for that, we sampled randomly pattern of connections by using random seeds, more than 1,000, to extract what we call a connectivity map, which are the average binarized connectivity map derived from the Human Connectome project. This sampling, we did it in two different ways. We did it based on random seeds, but we also did it based on stroke lesions, just because we're very interested in stroke. And you'll see that in the end, it had a little twist that actually turned into an advantage. So we had 1,333 stroke lesions that we used to extract a pattern of connectivity in the brain, and we had those random seeds that are not completely random because they match in size and brain side to extract this connectivity. And the profile of connectivity was characterized by using the MMP parcellation from datasets. So we have this big matrix of how things are connected together according to the seeds that we have. Then our strategy was to try to simplify those profile of connections using principal component analysis. And what you can see in this chart is that when you use stroke lesions, it's much easier to model pattern of connections than when you use random seeds. So that's mostly because stroke lesions do not happen randomly in the brain. And then when you look at the component of connectivities that you can extract and you try to correlate them with task-related functional MRI maps, you'll find that even if you correct for multiple comparisons, you have all your components that are always correlated quite highly with pattern of activation. So the component of structural connectivity seems to be matching the functional division of the brain. And you don't have that many components because for the stroke lesion, for example, you can extract more than 90% of the variance of the connection just with more than 33 components, if I remember well. While with a random seed, you'll need more components because they're really random. What is interesting is a relationship with function. We tried to compare whether random seeds were correlating better because they're more random and representative of the distribution of the connectivity of the brain with function, or if stroke lesions were correlating better with function, task-related meta-analytic map of function. And what we found is actually stroke lesions correlate better with functions than stroke lesions disconnection correlate better with functions than actually random seeds. And this means that the way we study functions is a little bit biased by the way we've been seeing patients and coming with stroke lesions in the hospital, complaining about not being able to speak or not being able to do things in everyday life, and neurologists and scientists like ourselves questioning ourselves about what is the function that is behind that, studying in patients and then trying to study it in controls and do task fMRI about it. And this little difference between the two might be the bias of exploration, what we can see in patients that have been influencing the way we explore function in healthy humans by means of fMRI. So somehow stroke lesions is a better way to seed tractography if you want to decode function. So what we were interested in too is like those components are interesting, but we'd like, we're brain scientists, we'd like to see a little bit of brain. So we run a high dimensional model where we've been able to extract for each component the contribution of each voxels of the white matter. And this gave us these beautiful maps here, which are like 4 maps that match 4 correlations that I showed to you before. For example, for the language, the component that correlated with language, we can see that the connections that are the most related to that component belong essentially to the arcuate fasciculus, so no big surprise here. When you speak about navigation, it will be more connection going from the visual cortex to the hippocampus and the retrospinal cortex, which are like critical area for navigation. And I guess the connection with vision is not a big surprise either. When you speak about eye field, you had these very strong connections between the two eye fields, and that does explain why when patients have a lesion in one frontal eye field, both of their eyes deviate. It's because they are highly interconnected in order to coordinate the orientation of sights. And when you look at the component that was correlated a lot with calculation, you can see that it is very localized, and this will match as well the work of Andreas Kleinschmidt reporting that the Gersma syndrome will be related to disconnection of a short connection in the depth of the intraparietal sulcus. And for those of you who don't know, the Gersma syndrome include symptom, which is acalculia, the inability to calculate. Those maps are very informative, but somehow we think like we cannot have that little number of components correlated with function because this does not cover the entire wealth of function as we know it. And so we thought maybe it's because some function emerge from the integration between the contribution of those different components, and our matrix of analysis is not well designed to capture this kind of interaction. And so the next step that we did, we flipped a little bit our matrix and tried to take the level of correlation between each component and each function in order to extract really voxels that will be in the white matter that will be contributing to any of the map of task-related activation from neurosynth. And this gave us a lot of maps that we summarized in this figure that you can see here, that is only showing the top contribution of the white matter to given terms. You can see clearly on the left hemisphere some areas of the white matter being dedicated to language-related tasks in fMRI, and part of the white matter in the right hemisphere more being dedicated to tasks measuring impulsivity and maintaining information. How statistically significant were these maps? If you analyze statistically the effect size of this result, you can see that all the mapping, with the exception of some part of the brainstem, was between a medium to high level of effect size. But the distribution is not the same in the brain, and there is clearly a very large effect size for arcuate connections. And another way also to explore those maps is to look at how many terms we load onto the white matter with a medium, more than a medium effect size. And you can see that there is a huge asymmetry between how many terms we load onto the left hemisphere compared to how many terms we load on the right hemisphere. What does it mean? Certainly not that the white matter in the right hemisphere is doing less than the white matter in the left hemisphere. What does it really mean, I think, is that we know so much more about the left hemisphere than the right hemisphere, and you have a strong asymmetry of knowledge and studies that are reporting activation in the right hemisphere compared to the left hemisphere. And what we just code here is a lack of exploration of function that will be in the right hemisphere. Overall, all this material is available for you to use and analyze, and if there is any questions or if you need any of those 3D models such as the average tractography, do not hesitate to write directly to me to ask me for this material. I'd like to thank all my collaborators and stress that we do have a special issue on brain parcellation and the relationship to function that is running in brain structure and function. You can send your papers. We will be accepting update in already published parcellation if you wish to submit it to the journal. It will be sent to review, and we will be happy to support your scientific effort. Thank you very much, and I look forward to your questions and your emails. Thank you.